Hello guys, this is my first YouTube video and I am going to teach you about the entry of HIV virus today. So let's start with the virus. This is a one HIV virus. So the first step to entry is the attachment and fusion step. So these are the receptors, CD4 receptors or chemokine receptors. So the virus will first attach to this receptor, and then there is a nucleocapsid. Nucleocapsid is basically the nucleus membrane surrounded the viral genome. This is genome. This is so this is a retrovirus. So it have a single standard RNA virus, and then then this nucleocapsid undergo to the uncoating step, and in this step the viral genome comes out. This is the viral genomic RNA. This undergoes to the reverse transcriptase. This enzyme is already present in this nucleocapsid. So now this virion genomic RNA undergoes reverse transcriptase. Then then after reverse transcriptase, then there is a cDNA and RNA complex which also undergoes to the reverse transcripted, and then it forms the double stranded RNA. Now this double stranded RNA, now there is also an enzyme called integrase. This integrase enzyme helps this double stranded is form double stranded RNA to integrate it into the host chromosome. The human chromosome is inside the nucleus. So this is the nucleus, and then here is the viral DNA. It is already integrated into the host chromosome. So now this integrated host chromosome undergoes to the transcription through the ribosome. The ribosome, which is present in cytoplasm outside the nucleus, so it undergoes to the transcription, and then it form two types of RNA. The first one is viral RNA, and the second one is viral mRNA. So what does viral mRNA do? Viral mRNA, this forms two types of protein. One is structural proteins and another one is regulatory proteins. The structural proteins helps to assembly and form the new variants, while regulatory proteins helps the viral to regulate and to further transcripted. So this protein helps ultimately helps viral vRNA or virus to assembly and to form new virus further. So this viral RNA, the first form viral RNA then undergoes assembly, and then this assembled RNA undergoes to the wording and release. So this virus is now coming out of the cell through budding and release. Then there is the process called maturation. This is the process where the protease, the enzyme protease, comes here comes so then this virus will mature into the mature variant. I will talk about these antiviral agents later. So first I will talk about the two types of HIV. There is HIV-1 and HIV-2. So HIV-1 is a worldwide pandemic, and the HIV-2, this is isolated in West Africa. So it have only one difference between HIV and HIV-2, that it causes AIDS much more slowly than HIV-1. So the AIDS, so if you have HIV-2, then you will have AIDS much more slowly than HIV-1. So otherwise these are all clinically similar. Okay, so this is the basic structure of HIV virus. So there you see a reverse transcriptase enzyme, and there is a protease enzyme which helps in maturation, and then there is integrase enzyme which helps you to integrate into host chromosome. So this is RNA. So yeah, you have two copies of single stranded RNA. Yes, this is retrovirus single stranded RNA transcript to double stranded DNA. Yeah, as you have seen earlier. So reverse transcriptase and this host enzyme. So yeah, so this is transmembrane envelope protein. We have this outer is called GP120, and this inner one is called GP41. So this helps into attachment. I will explain it further in the video. So So yes, let's see how HIV enters. 3 steps: binding of HIV via G120 envelope protein to CD4 receptor attachment. Yeah, so we have seen this earlier. Here this is G. So this is G120 envelope protein, and it attached to CD4. And this binding of co-receptor via conformational change, I will talk this about later in fusion of virus. So we have addition of CD4 receptors. We have another two main co-receptors which is targeted by HIV virus to enter into the cell. So these two important ones are CXCR4 and CCR5. So HIV variants mainly using CCR5. So with those HIV variants which use CCR5 more commonly than CXR5 are called R5 virus, and which using CXR5 are called X4 viruses. So let's see further. Okay, so we have when the virus enters the blood, it binds a protein on its surface GP120 to a CD4 receptor and co-receptors CXCR4 or CCR5 on CD4 shell.